Welcome folks. Happy Wednesday. How's everyone doing? How's life? How's it going? Good. Awesome. Awesome. Cool. So first I want to start with some logistical announcements. So hopefully, I'll pull this over. So on the Canvas homepage, the main things I want to highlight, I mentioned last time, but basically I'll be posting the recordings from this class onto this page, and I'll be posting the slides as well. I'm not done making slides. It's like very painful, but I'm almost there. So I'll be posting the remaining slides once I have them, and in general, after each lecture, I'll be posting the recordings here. So like this is the recording from Monday's lecture. These are just the normal Zoom recordings, so it's not going to be like the full classroom or anything, but I guess it's got the most important pieces. I typically try to post these within a couple hours after class, but sometimes meetings come up or I have to take the bus home or anything like that. So I promise you that I'll always post the recordings by the night of at the latest, so I'll never wait till the next day. But yeah, just kind of keep an eye on that. Other logistical stuff. So everyone seems to have, or at least the majority of people have seemed to access the Prairie Learn assessments perfectly fine. So it seems like a lot of people are making progress on Lab 1. Awesome, keep it up. And I realized not everyone might have Python experience. So if you came in with like Java experience from APCS or something like that, and then never took the intro like Python classes, some of the challenges that we have ask for some basic Python work. So if you want to just kind of read up on Python syntax, I wrote an online textbook for another class that it's free, it's publicly accessible. I have a link to it here on Canvas that I just added. Thanks for reminding me, Marcus. And yeah, basically you can kind of read up on Python syntax here. None of the stuff that we're going to be doing in this class is going to be super like computationally intensive. You're not going to be coming up with any like super crazy algorithms. Mainly just like using the logic that you already know from CSE 11 or 12, but just coming up with Python syntax to do loops, to do conditional statements, stuff like that. So yeah, definitely read through these if you want to just kind of quickly learn, but it's not very intensive by any means. It's just like learn the basics of Python syntax to be able to do those challenges. I think that's all the logistics. Oh, and so I got confirmation earlier today that the final will also be through the Triton Testing Center. So I believe you are able to register for both exams now. I would recommend doing it sooner rather than later just to get a good time slot. But yeah, both of them are going to be through the Triton Testing Center. I think both of them, they will give you a non-graphing calculator and a note sheet, like not a note sheet, like a scratch paper sheet. But I don't think they let you bring anything else in. So just be aware of that. As I mentioned last time, both exams are going to consist only of problems from the non-coding segments of the various labs, which I'm going to be posting here at this lab. I think I didn't have this last time, so I think I'd forgotten to do it. But basically, I created this bottom section called Practice Problems Non-Coding that throughout the course, as you do the labs, I'm going to put all the non-coding problems over here where you can randomly generate as many versions of them as you'd like. So you can like kind of keep solving these problems and then keep generating new versions of it as much as you want. So basically, if you're able to solve all of the problems on this page, not right now, but like as I progress through the course and add the rest of them too. So if you're able to solve all the non-coding lab problems with a pen and paper and with a non-graphing calculator, you're gonna get a perfect score on the final and on the midterm. So study up. And yeah, I think that's my spiel. So any logistical questions, any logistical concerns, comments? No? Anyone on Zoom? I'm monitoring the Zoom chat, so feel free. No questions? Okay, cool. So I'll pick up exactly where we left off last time. So last time we ended on this slide where I just mentioned there's a lot of interesting properties of a genome that are just generally of interest to bioinformaticians and also are going to be relevant for the little coding challenges that we have on Lab 1. Oh, someone asked in the chat, sorry, no cheat sheet, right? Yeah, so my understanding is the Triton Testing Center doesn't let you bring in a cheat sheet, I think. I requested a note sheet, but then they said that they give you a scratch paper. So I think they don't do cheat sheets, but I'm not 100% sure. But you can email them if you want to know. I'm cool with it if they're cool with it, but yeah. Okay, awesome. So yeah, so this slide, this is going to help you do some of the problems on the lab. And then these are also some problems that are going to be in the non-coding section of the lab. But yeah. Okay, So I guess before I continue with today's topic, are there any questions from the previous 3 tabs that people might have that they want me to just kind of quickly go over? Everyone's all good. Everyone's an expert on DNA sequencing technologies. You're going to make the next Illumina and cut me in? Yeah. Okay. Awesome. Okay, so basically, just to quickly recap what we talked about last time, I introduced the class. I talked about how phylogenetics kind of plays a role in an epidemic, and that was going to be like the framework behind which a lot of the lab assignments are going to be coming from. You're learning generic bioinformatics techniques that are applicable to humans, plants, viruses, bacteria, but just having viruses as the scope kind of gives it a little bit of cool context, and the data set sizes are much smaller. We talked about sequencing technology, so we watched the video from Illumina of how sequencing by synthesis works, how they generate paired end reads. So remember, one fragment of DNA, they're able to get 2 reads from either end of that fragment that came from the exact same molecule of DNA in the sample. And yeah, that's where we left off. Cool. Okay, so today I'm going to talk about viral molecular epidemiology. Again, this is going to be kind of one of those backgrounds that is going to motivate a lot of the labs. So the second bullet and the fourth bullet, they're not like directly tested, but they're just some interesting context to like give some meaning behind what you're doing instead of you just being given a bunch of arbitrary assignments to do. Okay, so viral molecular epidemiology, I first want to define the concept of molecular epidemiology. So molecular epidemiology, it's the integration of molecular biology into epidemiology. So epidemiologists, they're trying to track pathogens, they're trying to track different public health states. And molecular epidemiology is let's take molecular biology and use it to help them do that. And in modern times, because sequencing has become so cheap with the existence of Illumina sequencers and now the various long-read sequencers that we have as well, mainly molecular epidemiology is focused on how do we use sequence data, either of the pathogens, of wastewater, of whatever thing that we're able to collect, how do we use sequence data from those samples to study epidemiology? So some context, every pathogen has its own genome sequence. And if I sequence samples from patients, let's say like you all do COVID tests and I sequence the swabs that you do, let's say that in each of these bathrooms, there's like some wastewater collection thing. This is what we do on campus to track COVID. We can sequence whatever DNA or RNA we can collect from those samples, et cetera. And then we can then use those sequences to try to do things like identify what pathogens are in our sample. So maybe we want to test for like Mpox versus SARS-CoV-2 versus other viruses. We could try to see if there's any interesting mutations. So this was a big deal when Omicron happened. I don't know if folks remember like the Omicron outbreak. We thought that our bioinformatics pipeline was broken because like there was a big uptick that wasn't being reflected in the testing. And then like 2 weeks later, we found out, oh no, that was accurate. It's just that like you poop COVID faster than you test COVID. So we actually like pre-detected the Omicron outbreak 2 weeks later, and we can identify like what are the mutations that are associated with this outbreak, all that cool stuff. And then those identified features can then inform public health response. So things like, does the term, I guess maybe I'll just kind of define it. So with COVID pandemic, the way that the PCR, have folks heard about the PCR tests for SARS-CoV-2? So how does that work? Does anyone know? Yeah, go for it.
So they take like a sample and they run a PCR on. So they break it down to just the DNA and then they sequence it so that you can see the DNA. Well, you do a PCR, so you have the thing you're looking for, which is probably the COVID like marker sequence that we have.
Okay.
And then you run it against the samples that you got to see if it has, like there'll be like a band across that'll have the.
Perfect, exactly. So let me just repeat that for the folks on Zoom and just for everyone. So you said, okay, I purify the sample that I have, and then I have some type of way of targeting the thing I'm looking for. So in the case of SARS-CoV-2, I'll have what are called primers that are targeting various regions of the SARS-CoV-2 genome. And then you said I do a PCR experiment. Wait, what's PCR? Huh. Yeah, go for it. Exactly. So PCR is an amplification technique. Basically, we can run this, it's called polymerase chain reaction. It's a way of giving a small amount of, I think it works just on DNA maybe. And I think you reverse transcribe RNA, and then you have cDNA that then you can do PCR on. But yeah, basically I have some sample with DNA or like some cDNA from RNA. I can then run this polymerase chain reaction, this PCR experiment, to then replicate. But the PCR test, they're specifically doing PCR given primers that were designed from specific segments of the SARS-CoV-2 genome, and they're based on the reference genome that we have. So the reference genome being a representative genome sequence that we have for the sample, like for the species. So what ended up happening in the middle of the COVID pandemic is, so basically the way that you turn this into a diagnostic test is I have these probes targeting SARS-CoV-2. If PCR succeeds, this sample has COVID. If PCR experiment fails, this is a negative test. So success of a PCR experiment means I'd say this is a positive test. This sample has SARS-CoV-2. If it fails, that means it's a negative test. Well, how could it fail? One way of it failing could be maybe some lab tech accidentally screwed up the experiment. So even though there was SARS-CoV-2 in this sample, the experiment failed just because the bio experiment was not run properly. Another way that you can fail the experiment is like maybe the virus has mutated in a way that I was targeting these probes based on the reference genome, but if enough mutations happen in the region that I was targeting, that PCR probe is not going to be able to bind. And then artificially, the sequencing experiment is going to fail because it truthfully did not find the thing it was looking for. So in general, so kind of going back to this slide, the features that we can find in terms of like interesting mutations, if we see, oh, hey, I'm starting to see a lot of mutations in the regions that we're targeting with our diagnostic tests, maybe I should come up with another version of my diagnostic test. Or if we see, oh, hey, there's certain regions of the virus that are not mutating very much, maybe I should incorporate those into an updated vaccine strain. So like with the COVID vaccine, we had like the later iterations of the newer RNA vaccine that like then was targeting Omicron specifically. This is kind of how you do it. You basically find what are the mutations that are interesting, and then let's either design different diagnostics, different vaccines, all that good stuff based on those mutations. Okay, so what is viral molecular epidemiology? It's the use of, you can use the properties of evolution of the virus to study a viral epidemic. So some questions that we might want to be able to answer is things like, how did the virus spread across communities across the world? So if I collect all these different viral, like these COVID tests, basically, or whatever viral test I'm doing, each of those tests has some timestamp associated with it because someone like physically did a nasal swab, it got collected, yada, yada. It also has information about like the geographical region from where this person was. Maybe it has information about who this person was, like their age, their, any other metadata. So what I could do is just take a map of the world and like over time see how are the counts in these various geographic regions increasing or shrinking. I can do that purely based on positive test that I know the geographic region and the timestamp of that test. There's a question on Zoom. How do you predict mutations to fit your PCR test in the case of a failed test? Good question. So yeah, how do I actually, so this goes back to my previous slide that I mentioned. How do we actually adapt our test? Basically, you see a bunch of cases where it seems like the person really does have COVID. Either you do a blanket sequencing experiment or like they have all the symptoms and like maybe the other probes did test positive, but just one of the probes failed. And you just scratch your head for a long time and like, what's going on? Why is this failing? And then you realize like, uh-oh, there was enough mutations. Now let's design a new one. So it's usually not super well thought out because you don't really know it's broken until it breaks. And the fact that it breaks makes it hard to actually detect. So it's usually you have a bunch of suspicious like, some of them are failing to replicate. That's kind of the rationale behind having multiple probes is that like, hopefully all of them won't break at the same time. Hopefully only a couple of them will break at most. And then you can still kind of use the rest of them to figure it out. But yeah, basically very hand wavy is the unfortunate answer. Yeah, great question. So another question that we might ask is what transmission clusters exist in our population? And this was actually super important to ask during the beginning of the COVID pandemic. I think everyone here was probably like in high school during that at the very beginning. Yeah, so in the early days, there was questions of like, what do we shut down? Do we shut down restaurants? Do we shut down bars? Do we shut down travel? What do we do? So this plot here, I'm not going to go into super detail what it's showing, but basically this network, the nodes represent people that tested positive with SARS-CoV-2, and the links between them were determined purely based on similarities of their viral sequences. So they collected a viral sample from each of these patients, they sequenced those samples, and then they put links between them if their viral sequences were within some amount of similarity. And the directionality of these branches is based on time. So if this person's sample was collected before this person's sample, they just kind of infer that the directionality goes that way. So I want to emphasize real quick the structure of this network. So if you ignore the colors, just look at the structure. It was completely determined purely based on sequences that they collected from people. So no information about like who they were, what they were doing, anything like that. The structure was purely based on sequences, which we as bioinformaticians are able to analyze. Then once you question them about like, hey, what types of interactions were you having? When you do some type of follow-up like contact tracing, if you survey them and see, oh, all these people interacted at this bar or at this temple or at this wedding or local travel, then a public health official that's trying to make determinations of like, do we close off travel? Do we close off bars? What things do we close off? Can make a much more informed decision of like, oh, hey, maybe travel is not actually that bad. It looks like there was only one of these connections that was related to travel versus maybe bars were kind of a hotbed for SARS-CoV-2 transmission. So maybe we like limit how many people can go to bars, but maybe we're okay with some amount of weddings or yada, yada. So basically, instead of just kind of closing your eyes and trying to make guesses based on no information, we can actually get a lot of information just based on the sequence data, and then we can overlay additional metadata to be even more informed. But so I'll pause for questions, kind of makes sense. Everyone with me still? Yeah, go for it, yeah.
So can you just clarify what like the black dots and the red dots mean?
Ooh, good question. So the question was, can I clarify what the black dot and the red dot means? So I'm not 100% sure. My understanding is that they're related to, so I believe they were looking at a specific city. I don't remember what city it was. And I think imported source means someone came into the city or someone had traveled and then came back into the city. Versus local source, my understanding is someone was already in the city and then transmitted. But then again, how did they get it? Then depending on how far back you go. So, but I think local versus imported is like, were they already a resident here versus was there a very recent travel arrangement of some sort? Yeah. Great question. Yeah, go for it. Go for it. Ooh, good question. How do we determine if viral sequences are similar enough that there's a link? Hold tight until week 7. Yeah, we're going to talk about that. But so I have a CDC contract to build a tool called HIV Trace, which we do this for HIV molecular epidemiology. But basically, the short answer to kind of satisfy a little bit is, or maybe not satisfy, some higher knowledgeable person tells us this is the threshold that for this virus dictates if it's of interest. So the CDC for HIV defines 0.5% genetic distance between two viral sequences as being of high interest. And they define 1.5% genetic distance as linked, but not as high of interest. And how they came up with 1.5 and 0.5, like those are magic numbers that like probably just sounded good to someone. In practice, they had survey data that then they overlay with the genetic linkage to try to see out of the people that we know actually were related to each other, maybe they are known sexual partners or like somehow we know that these people were actually linked. What thresholds in this distribution of pairwise distances puts people that are linked together and puts people that are not linked together not together. But like anytime you hear 0.5 or 1.5, like that's a clean round number that someone arbitrarily picked. Yeah. And it varies like virus to virus, it varies region of the virus, all sorts of stuff. Okay, great question. Any other questions? And same for this. I think this they probably picked one mutation because this was SARS-CoV-2 and SARS-CoV-2 evolves much more slowly. Awesome. Okay. So that's one question. And again, most of this information is determined purely computationally using bioinformatics techniques. How is the virus mutating across the epidemic? So each of those samples that we collect, we can sequence them. We can determine what are the mutations in this sample. What mutations are in the sample? That's bioinformatically equivalent to asking what lineage is this, like Omicron, Alpha, Beta, whatever. So what I could do is just plot over time what is the prevalence of each of these different lineages in the population that I'm looking at. So then I can maybe see, oh, hey, this purple one kind of started overtaking the other ones. Maybe this is interesting. But then, this red one like really started growing. And oh, shoot, this pink one like really took off and is like super, super dominating. So maybe something's special here. So this might be able to inform some amount of like, are there any specific mutations that make it more transmissible, more infectious? What is going on here? Or maybe it's just purely an accident. Maybe it's some other confounding factor that like maybe there's like a big party I think this actually happened in the early days of the pandemic. There was like a big party at SDSU, and then like San Diego County just like upticked in COVID numbers like that week. So it might not even have anything to do with like the specific viral mutations, but just whichever virus happened to be at this big super spreader event happened to be the one that blew up. So at the very least, though, it gives you some insights into what might be interesting to pursue. Questions? All good? Okay. So another question that we might want to be able to answer is, what is the molecular mechanism by which the virus invades our cells? So we could theoretically sequence it, annotate that genome sequence, see what are the genes here based on those genes, and based on prior infectious disease knowledge that we have, what mechanism might be how this virus is specifically invading our cells. So SARS-CoV-2 was a coronavirus. Coronaviruses typically have these like coronas, these like they have a corona, like these little proteins on the outside that can bind to the outside of your cell, and then it has this way of like invading the cell. I'm not an immunologist, so I'm not very knowledgeable about that stuff, but basically something about this virus tells you this mechanism. And then once you can figure out what are the specific transmembrane proteins by which this virus is using to invade the cell, maybe I can come up with therapeutics that target various stages of this pathway. So maybe I can come up with some inhibitor that prevents things from binding to this receptor. Or maybe I can come up with something that detects these spike proteins and then like kills whatever it sees that has those. So somehow, if I can understand the mechanism, that can inform therapeutics, interventions, stuff like that. Oh yeah, go for it. Do you have a question? No, yeah, sure. So what if I want to know, are any of the mutations impacting the infectiousness of the virus? So the virus is going on for a long time. I want to see all these mutations are accumulating. What impact do I think they're going to have on how the virus functions? What I could do is actually take the three-dimensional structure of the protein. So this is the spike protein of SARS-CoV-2. And I can superimpose specific mutations onto the three-dimensional structure of the protein. So here, P323L, it stands for at amino acid 323 of the spike protein. P, which I think is it phenylalanine? Okay, I'm not very good at the chemistry side of bioinformatics. Bear with me. I think it's phenylalanine. Had a substitution into, I think, leucine, maybe, probably? Nodding? Okay, perfect. We got the chem people here. Awesome. So yeah, so this, okay, so at this position, what used to be a P mutated into an L. Remember, each amino acid has various chemical properties, various physical properties. I can see how do I expect that to impact the structure of the protein. I can do what are called molecular dynamic simulations to see how does this impact how well it binds to the receptors that it's trying to bind to. And then from that, I can make some inferences about what I think the impact on infectiousness or other downstream things might be. And then our goal for this class, which is going to be the motivation for all the subsequent weeks of this class, is how do we translate these questions into formal computational problems that we can theoretically solve? You said you took Pavel's class already? So formal computational problem is going to be something that's going to be resounding in your head forever. And I think, Marcus, that's probably you as well, huh? Yeah. Formally defined computational problems. So that's going to be the goal of this class. We're going to take a lot of these more abstract questions, turn them into computational problems, solve them. In this class, you're not going to be implementing too many of these things from scratch. You're going to be using whatever are the current best practice tools. And then when you get to Pavel's class, then you're going to implement some of them from scratch, and that's a lot of fun too. Yay. Awesome. Okay, so that's that section. I'm going to pause again and see if there are any questions, concerns about anything that we've talked about. All good? Okay, perfect. So the last section, this is just kind of a quick rapid fire of some Unix command line tools that I personally think, or actually, do I even rapid fire them? I don't think I do. I think I just kind of describe the Unix command line. So in general, any bioinformatics analyses that you're going to be doing, you're going to want to get comfortable with using the Unix command line. So in this class, we're going to have a bunch of Unix challenges that are going to help you practice. Week 1 is a little bit heavier on just practicing it for the sake of practicing. In practice, the data sets that you're going to be using are going to be much bigger than the ones that we're using in this class. So like a single human genome sequence experiment might have like over 100 gigabytes of data. One NovaSeq run, which is like a big Illumina sequencer, could have like between 100 to a terabyte of raw sequence data coming off of that thing. So in practice, usually what you're doing is you have like a smaller laptop that's way too small to actually run any of these. And most analyses that you do are going to be on some beefier server that you're remotely connecting into. So you really want to be comfortable using the Unix command line, just in general for your future careers. So the Unix command line, the terminal. So the Unix command line is critical for running bioinformatics tools. some tools have graphical user interfaces, but not very many. And also, in my experience, most of the tools that have GUIs are not very good. So the performant tools typically are from the command line. Also, most real analyses are too large for your laptops. You're probably going to be connecting to some headless server where you don't have any way of like running any graphical user interfaces at all. So yeah, you want to get comfortable using Unix both for this class and for future jobs. I have a couple links to some resources that I highly recommend people work through. One of them is this platform called sandbox.bio. It's a web platform that in your browser, they have a virtual terminal with all these little tutorials that you can work on. In my data structures textbook, I also have a section just about how to use the Unix command line. So I would also recommend referring to that. And then beyond that, I was going to spend the rest of today, because this is my last slide, I was going to spend the rest of today just kind of clicking around on the Unix command line, asking people if they want me to do any specific things, what they want me to practice on. So I'll go ahead and pause there again. This is my last slide. Any questions about anything I rambled on about for an hour and a half? Did you include last time? Question? Yeah, go for it.
Oh, there it is. My question is like for the testing, or for the exams.
Mhm.
So you said like whatever is in the practice will be on it. So for example, will there be any different variations? In like one of the questions we had, we have to determine like the coverage based on one position. But what if it's like here on this website, it talks about like the whole genome or one locus or something like that?
Ooh, good. So the question was on the exams. Is there going to be basically any expansions of any of the problems that you've been doing on the reading, on the lab assignments, such as like, so was it an article that I linked to?
Oh no, I just found it online.
Oh, I see.
Yeah.
So I'm guaranteeing you that every single question on the exam is going to be one of the non-coding problems from any of your labs. It's going to be identical to it, just a different randomly generated version of it. So it's good to be able to do the other versions of these problems as well. Like if you find some other resource that goes a little bit further, definitely learn that stuff, because I'm just giving you like barely dipping your toes in the water level of exposure. But for the exams, it's verbatim going to be the exact same problems, 100% guaranteed. You can quote me on it, I swear. Yeah. No surprises. And also no writing new exams for me. Yay. Awesome. Okay, any other questions about logistics, about any of the concepts I just talked about, or do people want me to jump over to the Unix command line and we hack around a little bit? Do it. Laptop? Wait, let's do it. Okay. So let's see. What data set should we use? Let me pull up my terminal. I'm going to make this a little bit bigger.
Are you going to show us some super confidential CBC data?
I cannot. I took an online training that told me not to. But I could, so I could show you sequence data. I was actually going to start with video game data because I like video games, but maybe not. Maybe I'll do sequence data.
No,
I'll say
this is sophistication, this is culture.
so. Okay, so we're going to start with some video game challenges that I have. So if you didn't know, in addition to bioinformatics, I'm a huge gamer and I am very obsessive about documentation. So I built this like open source database of a bunch of game information that I think is a good starting point for just like little practice with the Unix command line. So I'm gonna actually maybe let me stop sharing real quick. It's kind of hard for me to look in both places. So I'm gonna do duplicate screen real quick. There we go. Now people in the classroom can see my awesome Zoom backgrounds. I'm a huge Fast and Furious fan too. Okay, so and I'm gonna be changing my backgrounds every time. Let me actually just go down a little bit and let me hide this. Okay. So I'm going to first just type the command to make sure I did it right, and then I'll explain what I'm doing. Oh yeah, I did. Okay. So from the command line, one of the command line tools that I use all the time is a tool called wget. So wget is a command that you specify URL and it downloads it. So with wget, you can either specify -q, and maybe let me create a little temp folder. And so makedir is the command to make a directory. Hopefully folks saw that in like CSE 29 or equivalent. Oh, I already have one. CD changes directories. What's in here? Oh, whoops. Hide those. That would be a FERPA violation. Those are like project submissions from last quarter for CSE 100. So the wget command just downloads a URL. So if I do like wget and then this TSV file that has all the N64 games that were ever made, it has this lengthy description that like it just tells me the status of it connected to this location. It downloaded super fast. And then now, oh yeah, ls-l, and now you can see here is this file. So you might not be super comfortable with the various flags that you can give to the ls command. For me personally, I like to, and by the way, is the screen too low for people in the back, or should I like try to make it like the top half of the screen? Or can people in the back see? Okay, perfect. So for me, when I see ls-l and I see like 61656, I'm not good at in the top of my head like saying, is it 60 kilobytes? Is it 6 kilobytes? Like I'm always off by one. So -h is useful for, whoops, ls-lh. So ls on its own just lists the files. ls-l lists the files with additional information like the timestamp, the size. If you then pass -h, which I can just do in a single flag, but you could do separate flags if you want, it gives you a human readable file size. So here now it says instead of exactly 61,656 bytes, it's 61 kilobytes. So just a little bit easier for me to get order of magnitude. One command I use a lot is called less. So the less command just opens up a file in the terminal. It doesn't edit it. It just opens it read only, which is useful instead of using. So I use Vim for everything to edit files, but you don't want to necessarily edit a file that you're just trying to analyze because then you might accidentally make changes that you didn't want to do. So less is useful for just looking at the file. And in this case, you might notice that it actually wraps around by default, which might be what you want. Or it might not be what you want. So you could do less-s. So bless you. If you specify a capital -s, what it's going to do is not wrap around. So you'll notice now it opens it, and then, oh, there's a question in the chat. Type rm-rf/. No. You can't trick me that easily. I'm slightly smarter than that. Not by a lot. Okay, so you'll notice that now that I added the less-S, you'll see that there's these arrows on the right. This is telling me that there's more stuff that I can scroll through with left and right. I can also scroll down, I can scroll up. If you're comfortable with Vim, there's also just the standard Vim commands like capital G takes me to the bottom, lowercase g takes me to the top. If you're not comfortable with Vim, it's useful to be comfortable with it, but you don't have to. Yeah, so yeah, that's how I just open files. But you'll notice in that first command that I ran, let me just delete this file now. You'll notice the first command that I had written was wget -qo-. Okay, what's going on here? So wget -q means quiet mode. So when I ran wget, it showed all of this verbose progress information. But -q says hide all that information. Don't tell me anything. Just give me the contents. And then -O specifies where it's going to output the file. So I could do -O and specify some alternative file path if I wanted to. Or in this case, I did -O. So this is technically equivalent to this. I did -output is going to a dash. And the dash here for the output file means print it to standard input. So if I were to do that without this command at the end, so here wget quiet, so don't show the status updates. The output file, I want it to be standard output. And then here's my URL. We get this super long mess of stuff just printed to standard output. So what I can do, and this is something that you're going to really want to get comfortable with, is what's called piping. So in Unix, you can use a vertical pipe. So this is just like shift and then whatever the slash is that's above the enter button. So the vertical pipe symbol means feed the standard output from the previous command into standard input of the next command. So less, previously I had run it less-s and then I had given it a file. But if I don't give it a file, that tells it read from standard input. So what this is going to do is that big mess of stuff that got printed right now to standard output, it's going to be fed into standard input of less. And now I can just read it on the fly without having actually like saved it as a local file. Okay. Are folks with me? Questions so far? Yeah, go for it. Ooh, good question. What's the difference between less versus like head versus tail? So good question. And head and tail are two very common commands that I like to use as well. So less lets you open it in a way that lets you scroll up and down and left and right technically. So less I can kind of like keep playing around with it interactively. Head, it only prints out the top some number of lines that you specify. I think by default it's 10 lines. So if I just run head as what I pipe it into, it just prints the top few lines of the file. And I can change how many lines by specifying dash the number of lines I want to show. So head -2 is going to say, show me the first 2 lines from whatever I just piped in. And now it shows just the first 2 lines. Tail, on the other hand, does the bottom of the file. So if I do tail without anything, I believe by default it's 10 lines, but either way, it's the last some number of lines. And I can alternatively, again, specify how many of the bottom lines I want. And there we go. So this is actually very useful for bioinformatics because, like, let's say that I'm doing a sequencing experiment and I have like a 100 gigabyte file, but I want to just test my analyses on like a handful of reads. I don't want to do it on the entire like 10 or 100 gigabyte file. You could theoretically pipe it to head and then specify like, oh, I want to do it on, wait a minute, how many lines was each record in a fast queue? What was it? Anyone remember? 4 lines, right? So let's say I want to do it on the first 100 reads. I could pass it to head -400 and that'll, sorry, the first 100 reads, and that'll extract the first 100 reads where each read is 4 lines, aka 400 lines. Or maybe I want just the last 100 reads for some reason. So yeah, so head and tail are useful for that, but they don't let you scroll. They just kind of truncate a snippet of it, versus less is useful for kind of like interactively going up and down and left and right. There's a question in the chat. Will we need to memorize Unix commands for the exam since we can't have a cheat sheet? If there's any questions on the labs, which I believe there is, you have to be able to answer that type of question, I don't think with a cheat sheet. Triton Testing Center didn't confirm, but I believe you can't have a cheat sheet, I think. I'm not 100% sure. So I would just get comfortable knowing these. In general, I mean, kind of the whole point of this class is to get comfortable with these in the first place. So I think rather than thinking of it as like memorizing, like in practice, you never want to have to like, you want to get comfortable with these to the point that like you can start chaining these commands together really quickly. Because like the motivation here is it's a way of writing zero code. Like you're not actually writing a Python or C++ program to do this. And you can do very quick analyses just off the top of your head, which I'm going to show you on this data set. That's my instructor answer, I guess. Awesome. Okay, so let's do some actual analyses on this. And I can show you how powerful the Unix command line can be. So here, this is a TSV file. It's tab delimited plain text file, where each line is one video game in the N64. That's the Nintendo 64 for you youngins that were born well after its release. Are you all post 2000?
Yeah.
Oh, man. Oh.
I was born when the GameCube was dying, and I'm assuming so.
Oh, GameCube was like my childhood. Oh, that pains me. Okay, so N64, this is a relic of the past for all of you. You're learning history. I should market this as a history class too. Get some extra money. Okay,
But it's from the classic, so like, it lives in all of our hearts. So.
there we go. Okay, it's in all your hearts. Okay, perfect. That makes me feel a little bit less old. I'm sage. I'm not old. There we go. You're not supposed to laugh. I'm just kidding. So let's say I'm interested in seeing, so over here, so the columns are the identifier. This is a unique identifier that every game has. Then the title of the game, the developer, the publisher, the rating that the game had. So like E for everyone or T for teen, whatever. The region, so NTSCJ is Japanese. There's also NTSCU for the US. And the release date. So what if I want to know how many games were released in each of the different regions? And now that I'm a dinosaur, the N64 is pretty much totally dead. So no new games are going to come out. So we can be fairly confident this is going to be stable. So what column is the region column? Well, that's column 1, 2, 3, 4, 5, 6. So there's a command line tool called cut that you can specify which field you want to extract. And cut is weird, where everything that you've been learning about computer science is 0 indexed, but cut for some reason is 1 indexed. So the first column is 1, second column is 2. I think I said 6, right?
Yeah.
Okay, so I want to extract the 6th column. So I want to do Oh, did I say 7?
No, it's 6, it's 6. I counted myself at 6.
Okay, it's 6. Whew. Okay. So I can say I want to cut the 6th field from this file. This is going to be relevant when we talk about read mapping formats because those are going to be tab delimited. So next week when we talk about the SAM format, this will be helpful. But I want to extract the 6th column. You could alternatively specify which delimiter you want. So in this case, by default, it assumes tab delimited. But if I had like a CSV file, I could specify comma delimited or some other weird delimiter that I want. But in this case, it assumes by default tabs, which perfect. It successfully extracted that column that has all the regions. Okay, well, region is not a region. Region is the header of this. So it would be nice if I could omit just that line. So one way to do that, I always forget how I can just remove the first line, but one way that I could do that I do remember off the top of my head is there's a tool called grep that will filter for any lines that match some specific pattern that you specify. So here, I want to match the string region. And as you get a little bit better with grep, there's additional things that you can specify. So region was the very first word of the file. So maybe there's like some region that has the word region in the name. That would be kind of unfortunate. So I could specify this vertical caret, which means beginning of file. And I can specify a $sign, or not beginning of file, sorry, beginning of line. And I can specify $, which means end of line. So when I didn't have those two, this will match any lines that has the string region, even if it's in the middle of the line or anywhere in the line. Adding the caret at the beginning will only match the lines that have region at the beginning of the line. So it has to be start of line, then the very next letter has to be R, then E, then G, then I, then O, then N, but there could be more stuff afterwards. $sign means end of line. So this will only match lines that start with the word region and then immediately end. No other characters. Okay, so if I run it like this, it's not going to be the desired outcome. What this is going to do, it's going to output the only line that had the word region. But I wanted to do the opposite of that. I wanted to get all the lines that don't match this pattern. So grep, you can specify -v for inverse. So -v for inverse. And now I get all of the regions without the string region at the top. So this is the data set that I want. Okay, so now I have a bunch of lines that just have a bunch of different, so for each line corresponds to one game, and it's the region of every single game that I have. But now I want to count the unique items. So there's a command that we're going to want to use. It's not going to be useful yet, but I just want to give a sneak preview first, called unique. So unique returns just the unique consecutive lines. So if you have like A, B, B, A, and maybe I can actually write that. Let me do that. Dash capital E maybe. E, or let's say A. So A, B, B, A. Does that do it? Nope, shoot. Lowercase e, oh, there we go. Okay. Oh yeah, okay. So imagine that I have A, B, B, A. If I pipe that to unique, what it's going to do is it's going to collapse all consecutive lines that are identical. So that'll return A, B, A, because it's going to collapse these two into a single occurrence, collapse these two into a single occurrence, collapse these two into a single occurrence. But that's not quite what I want on my original example. Because in my original example, I just want those single unique things. I want just one occurrence of NTSCJ, one occurrence of NTSCU, one occurrence of PAL, ideally with their counts. Which, by the way, let me go back. With unique, there's also a -C flag that you can specify. So C for count, in addition to just outputting the unique consecutive lines that it collapsed on each other, it'll output how many occurrences there were that it collapsed. So it says A2, B2, and A there was also 2. But it would be nice if these were all consecutive. Like I wish I could merge this with this somehow. So how might I do that? Just to make sure folks are still with me and not sleeping or anything. So just conceptually, without thinking of Unix command lines, what would be a technique that would get all the A's together? Maybe you first, then you. Yeah.
Sort it first.
Ooh, I could sort it. Is that what you're gonna say? Yeah, perfect. So if I had sorted my original input, that would have been really nice, because then all the A's would be next to each other, all the B's would be next to each other. And then let's see in the chat, did he say sort? Perfect. And then unique-c would have outputted exactly what I want. So in this example, what I could do is first pipe to sort, maybe I'll remove the unique-c for now. So sort is a Unix command that will just sort the lines of its input alphabetically in increasing alphabetical order. It's alphabetical, so I'm opening the man page right. And by the way, in general with Unix, man is a very useful command. It stands for manual. You can type in any Unix command after man, and it'll show you the man page, the documentation for that Unix command. So it can actually do a lot more stuff. So by default, it's case sensitive, but you can have it ignore case. You can have it ignore leading blanks. The one that I use a lot is numeric sort. So again, it's alphabetical by default, but if I have like 100 and then the number 2, I don't want 2 to come after 100. I want 2 and then 100. Human numeric sort. So if you did ls-l to get these human readable sizes, it could sort them as well. So basically read through this. There's a lot of useful things. For our purposes though, alphabetical sort is fine. Whoops. And then now unique-c gets us what we want. So let's go back to our original video game example. Okay, so these are not sorted. Let me sort them. And now, yay, I have a bunch of NTSCJs, a bunch of NTSCUs, a bunch of PALs. This is like Europe and Australia. And now I can do unique-c, and it tells me, oh, there was 199 games that were released in Japan, 296 in the USA, 274 in PAL regions. Success. So the reason why I'm getting this across is, in this case, we did it step by step slowly, but once you get comfortable with these, you can just kind of like whip out these types of things super quickly without writing any actual lines of like Python or C++ code. And the good thing about these is these Unix commands are very, very, very highly optimized. Like, they're much faster than anything any of us are going to be able to write, unless you're like some super expert systems programmer or something like that. So yeah, so that's kind of the motivation here. Okay, we have like 1 minute left. Any last questions or anything that folks want me to do real quick? Yeah, go for it.
Can you get your expert opinion on today's Nintendo Direct?
Ooh, today's Nintendo Direct. So I'm a huge Final Fantasy VII fan, if you read the syllabus. So FF7 Remake for Switch 2. Oh. Also the FromSoft game was kind of surprising, like the Bloodborne 2 looking thing. Yeah, it was a good Switch 2 Direct. I approve. Yeah, the pricing is, yeah, not great. Okay, well, yeah, we're basically at 50 after. Thanks so much, folks. Have a good day in lab. Make sure to go to lab. Marcus is dying to help you all with your lab, so please bug him. He's getting paid either way, so make him work for that money. And yeah, I will see you all next Monday. Thank you. Any questions from folks on Zoom before I end the Zoom call? Feel free to type in the chat. Feel free to raise your hand if you want to unmute. Yeah, no problem. Yeah, thanks so much, folks. Have a good rest of your day.